Alright guys, today we're going to do a quick overview of our E. coli bacterial transformation lab using the pGLO plasmid. You guys have already completed a little bit of the pre-lab, but I'm going to go over with some of the basic stuff with you also. So what we're going to start with is we're going to start by reviewing what we are talking about when we're talking about a bacterial transformation. And what we have to remember is that there was an experiment we talked about where Frederick Griffiths was studying different forms of pneumonia. There were two types of pneumonia cells. We had what we called S cells and R cells. And the S cells were what we call virulent. They were deadly and they did kill mice. And our R cells were non-virulent, meaning they were pretty harmless and they did not kill the mice. Now, what happened when Frederick Griffith mixed together heat-killed S cells with living R cells and injected them into a mouse is that that mouse died. So what we ended up with when we mixed those two things together was a dead mouse. And what he figured out based on this experiment is that something was transferred from those heat-killed S cells to those living R cells. What he didn't know it at the time was what that thing was DNA, but he called that transfer of material a transformation. Okay, so when we're talking about what a transformation is, we're talking about a change in the genotype and the phenotype of a cell due to it taking in or taking up some type of foreign DNA. And that does need to happen through the cell membrane, which makes it kind of difficult because remember, our cell membrane has both a hydrophilic region facing the outside and a hydrophobic region on the inside, and it makes it really, really hard for some of our polar molecules like DNA to pass through. So what we're going to do in our experiment is we're actually going to complete a bacterial transformation. And the way we're going to complete that transformation is by using a plasmid. Now, our plasmids are going to be small circular pieces of DNA that we find naturally in a lot of different types of bacterial cells. Now, what's interesting is that that small piece of DNA is found outside of the bacteria's normal chromosome. Which is kind of cool because that means that those plasmids can replicate on their own in a lot of cases. Now, those plasmids usually carry some types of genes that the cell doesn't always need to express, but it can express it in specific situations. So when we look at our E. coli cell in this picture I drew here, we have right here our bacterial chromosome, which again is going to be a circular piece of DNA. Bacterial chromosomes are circular compared to eukaryotic chromosomes, which are linear. All right, and then outside of that chromosome, we're representing them with pink dots. That's going to be our plasmid. And in the picture that we're showing right here, we're showing a bunch of plasmids on the outside of the cell. That's what it'll actually look like in our experiment. So what does our plasmid actually look like? Well, we're not using the same kind of plasmid that we sometimes find in our eukaryote or in our bacterial cells. We're using recombinant plasmids, and those are going to be plasmids that we have actually modified and inserted foreign DNA into. I'm not going to talk about how we insert that. We do use restriction enzymes. That is on page 413 of your textbook. You should review that process. Okay, so on that plasmid, we have modified it or edited it to include a few different things. The first thing that we have put on there is going to be this BLA gene right here. And what that gene does is it codes for something that we call beta-lactamase. It ends in ASE. So we know that that is an enzyme. And what that enzyme does is it breaks down a specific type of antibiotic that we call ampicillin. Okay? Now, what we do when we put this gene in here is we make it so that our E. coli that have taken in the plasmid, they can actually grow on a medium that has ampicillin in it because it produces that beta-lactamase enzyme that can actually break down the antibiotic. Now, the other things that we've inserted onto our plasmid are going to be the ARAC gene, the pBAD gene, and then the pGLO gene. Now, the ARAC gene and the pBAD gene, those are both parts of the arabinose operon, okay? And if you remember your operons, this is very similar to our lac operon. Now, what this does is it usually is going to be involved in the breakdown of arabinose sugar. Remember those words that end in OSE? Those are going to be our sugars, okay? Now, just like our lac operon, right, we are looking at breakdown or a catabolic reaction, which means that this operon is an example of an inducible operon, which means that typically it is turned off or we are not expressing those genes. But when arabinose is present, it is going to be on. Now, what we've done with this operon that usually has those genes for the breakdown of arabinose is we've replaced those genes that break down that sugar with our pGLO gene, okay? Our pGLO gene is going to produce something that we call the GFP, or green fluorescent protein, okay? And what that does is it makes our bacteria glow a nice bright green color under some UV light. So if we look again at what's happening here, okay, we have our ARAC gene. It's coding for this repressor protein. Okay, so through transcription and translation, we're making a repressor protein. Now, when there's no arabinose in our cell or in our medium, that ARAC repressor is going to be bound to the operator. When that ARAC is bound to the operator, our RNA polymerase kind of gets stuck on that promoter. Remember, the promoter is the binding site, and we cannot transcribe and translate that GFP gene. Now, if we do have arabinose in our medium, try that again. All right, if we do have arabinose in our medium, that ARAC repressor is going to leave the operator, and then our RNA polymerase can come through, kind of get onto that promoter, attach there, and we can transcribe that GFP gene and we can express it. Okay? So again, what's really going on here is that we're trying to get this plasmid into the cell. So when we look at our procedure, what we're trying to do is take this modified recombinant plasmid and get it to enter our E. coli cell. Now, when we're looking at a procedure, what we're trying to do is induce competence. So our entire procedure is about making our cells competent. And when we talk about what it is for a cell to be competent, it means that it can actually take up DNA. Okay, or take in those foreign plasmids. Now again, that's kind of a difficult thing because DNA is a polar molecule. It's pretty large, right? We know that. And our cell membrane, right? If we remember what it kind of looks like, it's that lipid bilayer. The outside is polar, and the inside is nonpolar. And we should remember the only things that really pass easily through there are going to be our small nonpolar molecules. So what we need to do is we need to do something to our E. coli cells to get those cells to take in the DNA. And the ways that we actually induce competence in our procedure is through a heat shock, in the presence of calcium chloride. Okay, so we're going to use those things, and what that's going to do is it's going to relax the cell membrane. And when that cell membrane relaxes and kind of opens up a little bit, we're actually able to get the plasmid in there. If we get the plasmid into our cells, we will transcribe and translate the different things that are on the plasmid depending on what's in the environment, including our green fluorescent protein, and we'll also make that beta lactamase. Okay, so how do we figure out which of our E. coli cells have taken up the plasmid and which ones have not? We're going to plate our bacterial cells on different types of mediums. So what you're going to see in your lab procedure is that you are going to plate on 4 different types of plates. You're going to have 2 plates where you're going to put the E. coli that you've exposed to pGLO, and you're going to have another 2 plates where you're going to put the E. coli that you have not exposed or not given that pGLO plasmid. And what we have in these plates are abbreviated, and I'm going to walk through those abbreviations with you guys. LB stands for Luria broth. Luria broth is just something that we put in the medium that has nutrients and all of the stuff that our bacterial cells really need to grow. Another thing that we see is that AMP. That AMP stands for that antibiotic we talked about a minute ago. That is our ampicillin. The last thing that we see is ARA. We only have that in one of our plates, and that is our arabinose sugar. So for your job today, what you're going to do is you are going to predict the types of things that you're going to see on these plates. The E. coli plus pGLO plates that have LB and ampicillin, or LB ampicillin and arabinose, and the E. coli minus pGLO plates that have LB ampicillin and LB. If you guys have any questions, feel free to